She graduated from Adnan Menderes University in Turkey with a bachelor's degree in plant protection. She graduated top 10 in her class. She worked with olives and wine grapes primarily. That's not working well. And she's working with my advisor as well on population genetics of the walnut twig beetles. So I'll let her go with her presentation.
Yeah, it's working. My project is rapid molecular detection and population genetics of Pityophthorus juglandis, a vector of 1000 canker disease in Juglans species. And what am I gonna tell today about my project? First, 1000 cankers disease, host Juglans nigra, and fungal pathogen-Geosmithia morbida, vector Pityophthorus juglandis, my objectives, materials and methods, and little bit my current progress. So 1000 canker disease is a complex disease which has fungal pathogen-Geosmithia morbida and the vector insect Pityophthorus juglandis. And then why it's named 1000 cankers disease? Because of formation of a large number of coalescing cankers around galleries and exit holes. That's why it's named 1000 cankers disease. And little bit history of 1000 cankers disease. The Western US 1990s severe decline of Juglans nigra was recorded. And then 2010, Tennessee Knoxville recognized 1000 canker disease. And after that, 2013, Italy has 1000 canker disease. And the current situation, 7 different states has spread. 1000 canker disease has spread to 7 eastern states, which is native range of Juglans nigra. So what are the symptoms of 1000 cankers disease? As you see, the yellowing leaves, crown thinning due to branch dieback, and then wilting, as you see. And this is really good example of crown thinning. And then this is my pictures. Just it was taken between 2 months before and after. As you see, yellowing and branch dieback, it's really clear to see. It's one of my collection sites. So the first picture shows the numerous of exit holes and entrance. And then the second picture shows small canker area under the bark. So it is hard to see because there is no clue outside of the tree. So why do we care 1000 canker disease? Is that really important? Of course, let's see a little bit facts. Currently estimated value of standing Juglans nigra is $568 billion. And then Juglans nigra can be used for timber harvesting, furniture manufacturing, nut production, recreation, and food source for wildlife. And then the wood, lumber, and logs are exported to 45 countries. So these all facts make Juglans nigra is really important, and then we really care. Fungal pathogen, Geosmithia morbida. Geosmithia morbida species are actually saprophytic, but Geosmithia morbida is the first phytopathogenic species reported in this genus. And then Geosmithia morbida can live in symbiosis with its vector, Pityophthorus juglandis, on Juglans species. And it's thermotolerant pathogen. It's almost 41 Celsius. It means 106 Fahrenheit. And higher temperatures resulted in small canker development. So the first picture shows under the bark canker development, and then second and third 4-week-old Geosmithia morbida cultures, and these are conidia, cylindrical to ellipsoid, and then conidiophore forming chains. So what has been done about Geosmithia morbida? Geosmithia morbida is genetically variable, but morphologically and ecologically homogeneous. And 5 genetic clusters with evidence of gene flow and high haploid genetic diversity was found from Italy and US. And Geosmithia morbida has lack of evidence of sexual reproduction. And this is really important. Previous research suggests pathogen and vector co-evolved long before the first report of disease. Pityophthorus juglandis, it's almost 2 millimeter in beetle, and then it's fluid feeding, yellowish to reddish color, And then the adult one create horizontal galleries, and then the larvae one can create vertical galleries. What has been done about Pityophthorus juglandis? High population of Pityophthorus juglandis are required for severe tree decline. High levels of genetic diversity and evidence of two genetic lineages among Pityophthorus juglandis were found. And evidence of two morphologically indistinguishable species were recorded. So two major haplotypes of a single lineage are responsible for the spread of this disease. And then neighboring state, Madrin-Sky Island and west of New Mexico, has the highest level of genetic diversity for the different lineages. And there is no treatment for thousand canker disease. And then the result of thousand canker disease could be devastating both economically and environmentally. That's why my research focusing on. My objective one is evaluate genetic diversity, spatial structure, and distribution of Pityophthorus juglandis from populations in the United States and Europe using macroslides. And my second objective, to develop rapid molecular detection of thousand canker disease. For objective one, my population structure of walnut twig beetle, materials and methods. Specimen collection, Pityophthorus juglandis will be collected from US, and then we are going to have DNA from Italy. And after that, morphological confirmation will be performed, DNA isolation and PCR. And then the last thing, coaxial capillary electrophoresis system will be used for my data analysis. And sampling. Multiple funnel traps will be baited with the lure containing male-produced aggregation pheromone. And lures will be replaced every 2 to 3 months. And traps will be checked twice a month. And then for Tennessee, we will locate our traps, 5 different counties. And in 5 different counties, we are going to select 3 different sites. So in this case, we are going to have 15 different populations from Tennessee. So how about the outside of Tennessee? We are going to have, for each state, we are going to have 3 different populations per each county. So this is one of my 2015 collection day. I was totally homeless at the end of the day because it took almost 7 hours in a day. And then we had flat tire. Life is hard, especially for graduate students, double hard. But we were good to go. So for my data analysis, freely available population genetics packages will be used. Let's move my objective to rapid molecular detection. Juglans nigra branches will be collected from California, Missouri, and Tennessee. California and Tennessee are highly affected places for thousand cankers disease, and Missouri will be used as a control. DNA isolation and PCR will be performed. Data analysis, again the same coaxial capillary electrophoresis system will be used. And then after that, for statistical analysis, R will be used. So from California and Tennessee, highly infected areas, and Missouri as a control, non-infested area. And then each place will be, from each place, 40 different branches will be collected. And then 5 to 8 diameter, 35 to 45 centimeter length. And then sampling. Each wood pieces, 10 different possible cankers area will be checked. And then after that, drilled and taken samples. And then including this, 5 opportunistic drills will be taken. And then my current progress of population genetics. As I mentioned before, I'm gonna have 5 different counties from Tennessee, and then 5 different counties will have 3 different sites. So in this case, I'm gonna have 15 different populations from Tennessee. And this is my current progress of population genetics study. And then we finished 2015 collection season. So this is my current situation. So 4 different counties, and I have moderately good results. So other parts of the US, I have samples from Pennsylvania, Ohio, and Indiana. Current progress of rapid molecular detection. The collection was done from Tennessee, Missouri, and California. And then each 40 segments, the samples were taken. 600 samples from California and Tennessee, and 400 samples from Missouri. And then I'm in the middle of DNA extraction, almost done. And then thank you so much. First of all, I would like to thank my country because they support me, and then my committee members, my collaborators, lab, and colleagues.